Welcome to Lecture 4.2, in which we will discuss the different types of transport that can be used to cross the cell membrane. This video over here shows a modified version of glucose that fluoresces green being taken up by cells. By the end of the video, you should have an idea of how glucose is entering these cells. Our learning objectives for this video are to differentiate between the different types of transport that are used to cross the cell membrane, which include passive, active, and bulk transport, describe the transport proteins used for passive and active transport, and understand how cells would behave in hypertonic, isotonic, and hypotonic solutions. First, let's define what a concentration gradient is. A concentration gradient is created when there is an unequal distribution of the concentration of something, such that there is a high concentration in one area and a low concentration in another. For example, if I drop some coloring into a glass of water, there is a higher concentration in the area where I dropped it, and a low concentration everywhere else. The direction of the concentration is often represented by a triangle, with the base representing high concentration and the tip representing low concentration. Over time, substances will naturally move down their concentration gradient, which means that they move from an area of high concentration to an area of low concentration. This continues until equilibrium is reached, which is when the concentration is equally distributed. For example, in the video, after the coloring is dropped, it begins to spread out in all directions where there was no color. Going down the concentration gradient is passive transport, which does not require additional energy. On the other hand, going up the concentration gradient does require energy, and is called active transport. Now let's talk about transport across the membrane. Simple diffusion is passive transport in which a substance is able to diffuse across the cell membrane without help from a transport protein. As discussed in the previous video, this would be the case for small nonpolar molecules and for gases such as oxygen. Here we see the phospholipid bilayer and oxygen molecules on both sides. The E stands for extracellular and C stands for cytoplasm. There are more oxygen molecules on the extracellular side in this case, and we can depict the direction of the concentration gradient with a triangle. Now these oxygen molecules will diffuse across the cell membrane. Since the movement is ultimately random, oxygen molecules can move in any direction, but there will be a higher net movement of oxygen molecules from the extracellular side to the cytoplasm side until both sides have an equal concentration of oxygen. Facilitated diffusion is a form of passive transport in which a substance diffuses across the cell membrane with the help of a transport protein. In other words, the transport protein is facilitating the diffusion of the substance across. This occurs when the substance is unable to cross on its own, such as if it's charged or is a large molecule. Charged ions use transport proteins called channels to cross the membrane. For example, potassium ions usually have a higher concentration inside the cell than outside. The potassium ions would want to move through the channel until they reach equilibrium. Channels are able to be closed or open, and when open, they can allow many ions through at once. For macromolecules like glucose and amino acids, carrier proteins are used. Unlike channels, these proteins have to bind to their substance and essentially carry them to the other side, which limits how quickly their substances can travel across the membrane. In this example, we have glucose represented by these hexagons, with the concentration being higher outside the cell. The carrier protein has to open up, bind to glucose, bring it to the other side, then release it. Osmosis refers to the passive transport of water across the cell membrane. Water goes through transport proteins called aquaporins, which are literally a pore or a hole inside the cell that water can flow through. Like all things, water would flow from an area of high concentration to an area of low concentration. A high water concentration means that there is a lot of water and that a solution is very diluted. Therefore, a high water concentration means a low solute concentration. On the other hand, a low water concentration means that there is not a lot of water and that a solution is concentrated. This means that there is a high solute concentration. This diagram is a good example. Here we have a beaker filled with water, represented by pink, and some solutes represented by purple. The two sides of the barrier are separated by this red dashed line, which represents a semi-permeable barrier that allows water through, but does not let any solids through. Only water can move across the membrane. The left side is more diluted, since there is more water and fewer solutes. The right side is more concentrated, with less water and many more solutes. Water would flow from the left, where there is more water, to the right, where there is low water. This also means that it is flowing from an area of low solutes to an area of high solutes. The end result is that the water level on the right side becomes much higher so that the overall concentration on both sides is about equal. Now let's talk about tonicity. Tonicity is the concentration of solute in a solution. Hypertonic, with hyper meaning higher, means that there is a high concentration of solute in the solution. In this diagram, this circle represents a cell, and there is a higher concentration of solute outside of the cell than inside. This means that water will want to leave the cell and enter the solution. Isotonic, with iso meaning same, means that there is an equal concentration of solutes in the solution and in the cell. In this case, water will still flow in and out, but the amount flowing in will match the amount flowing out, so there is no net movement of water. Hypotonic, with hypo meaning below, means that there is a low concentration of solute in the solution. This would cause water to go from the solution to the cell. Here we have a diagram of how red blood cells behave when they are in a hypertonic, isotonic, or hypotonic solution. As discussed, water would leave the cell if it is placed in a hypertonic solution. This causes the cell to shrivel up. When a red blood cell is shriveled up, it is said to be crenated. In an isotonic solution, there would be an equal exchange of water, so the cell would retain its proper shape. In a hypotonic solution, water would flow into the cell. The water entering the cell causes it to swell up, and one has swelled up to the point where it has burst. This red blood cell is hemolyzed, a term that refers to red blood cells lysing or bursting open. Plant cells are different due to the presence of their cell wall. When water leaves in a hypertonic solution, this causes the cell to shrink. Since the cell wall is rigid, this means that the cell's plasma membrane pulls itself in away from it, leading to it becoming plasmolyzed. In an isotonic solution, the plant cell is said to be flaccid because the membrane is still slightly pulled away from the cell wall. When water enters in a hypotonic solution, this causes the cell to swell up. However, unlike the red blood cell, the plant cell does not burst open. Instead, it becomes turgid, which means swollen or inflated, staying intact thanks to the rigidity of the cell wall. Now let's talk about active transport. Again, active transport occurs when energy is needed to move something up its concentration gradient. Primary active transport refers to when ATP is used as a direct energy source. This is generally done using transport proteins called pumps. The most famous example of a pump is probably the sodium-potassium pump, which moves both sodium and potassium up their concentration gradients. Sodium is generally higher in concentration outside of the cell, while potassium is higher in concentration inside the cell, as depicted by these triangles. The sodium-potassium pump starts off with being open to the inside of the cell. ATP, the source of energy, will bind to the pump, and this will allow sodium to enter the pump. The ATP will be converted by an enzyme into ADP, and this will allow the pump to move sodium to the other side, up its concentration gradient. Potassium will then enter the pump and be transferred up its concentration gradient to the inside of the cell, and then ATP combines so that the process repeats. The sodium-potassium pump essentially helps maintain this concentration gradient in which sodium is high outside and low inside, and potassium is low outside and high inside. This concentration gradient is essential for life. Secondary active transport does not rely directly on ATP, but on the concentration gradient created by primary active transport. When there is a big difference in concentration, a lot of energy can be harvested from a substance going down its concentration gradient. This energy can then be used to power the transport of another substance up its concentration gradient. For example, the sodium-potassium pump helps ensure that there is a lot more sodium outside the cell than inside the cell. This other substance also needs to go into the cell, but as shown by the triangle here, its concentration is higher inside the cell, so it would be going up its concentration gradient. The energy created by sodium going down its concentration gradient can be used to bring the substance into the cell. Since the two substances are moving in the same direction, the transport protein is referred to as a symporter. You can think of sim as similar or same. If the two substances are moving in opposite directions of each other, then the transport protein would be called an antiporter, with anti meaning they are the opposite of one another. Bulk transport is when substances are moved in or out of the cell through vesicles. Endocytosis is the process of taking particles into the cell through vesicles, with endo meaning inside and cyto meaning cell. There are three forms of endocytosis. One is phagocytosis, with phago referring to eating. This occurs when a cell engulfs a large particle in its environment. The other is penocytosis, with peno referring to drinking. This occurs when a cell takes little sips of its extracellular environment. Third is receptor-mediated endocytosis. This form of endocytosis involves a receptor that recognizes a particle, leading to a coated vesicle being formed. Exocytosis, with exo meaning outside, is when vesicles fuse with a membrane to release their contents out of the cell. This is generally how hormones and neurotransmitters are released. Constitutive secretion refers to exocytosis that occurs continuously, while regulated secretion is when exocytosis occurs in response to a signal. For example, neurons often receive signals that tell them to release their neurotransmitters. In this video, we learned that nonpolar molecules and gases cross the cell membrane by simple diffusion, ions cross by facilitated diffusion using channels, and macromolecules cross by facilitated diffusion using carrier proteins. We learned that water crosses a cell membrane through osmosis, leaves the cell when in a hypertonic solution, and enters the cell when in a hypotonic solution, and that this is done through aquaporins. We also learned that primary active transport directly uses ATP, while secondary active transport uses energy from an established concentration gradient, and that substances can enter or exit the cell in bulk through vesicles in the processes of endocytosis and exocytosis. That's it for this video. Thanks for watching.